Delighted to welcome you to our 10th webinar of the year. Before I welcome the presenter today, I just want to introduce the British Society of Soil Science, who are the hosts of today's webinar. So we're an international membership organization and charity, and we're committed to the study of soil in its widest aspects. And you'll see that within the webinar. We bring together those working within academia. We've got a growing membership among professionals implementing soil science in industry and government, and those with a keen interest in soils too. So just before we begin, some basic housekeeping. There's quite a few of you on the call today, so all your microphones have been muted. We're going to be taking questions during and at the end of the presentations, but this will all be through the question box. So please submit any questions you have by the wrap up of the talks, and this will allow us to get through as many as possible. So today's presentation has been awarded BASES and NRO SO CPD points. So if you're registered with either body, please contact us directly after the event. And also please be aware that we're recording today's presentation as well. So a few hours ago, unfortunately, we had apologies from Charlotte Chivers. She's suffering that autumn bug that's affected a lot of people. So she's not able to talk today, and she was going to talk about citizen science and its involvement within soil health assessments. We are joined by Calum Preece from Elementar, and Elementar are also the BSSS Corporate members who are helping to sponsor today's webinar. So they're manufacturers of high-performance analysis of organic and inorganic elements. They incorporate more than 125 years of experience in the development and manufacturing of high-class analytical instrumentation. So they've got loads and loads of innovation within this. Elementar is a well-known brand amongst many soil scientists, and their products are there to continue to advance our science across agriculture, chemical, environmental, energy materials, and other markets in many countries around the world. So we're delighted to be working with them today, and also to have Calum from Elementar to also present for us. So Dr. Calum Preece is a product manager for the environment market at Elementar UK. He's highly experienced as a scientific researcher as well. He focuses mainly on ocean sediments, but they're not a million miles from soils. And he's got a lot of expertise in biogeochemistry. So he's going to offer a commentary on various topics related to soils, as well as water quality and treatment, and also waste pollution and oceanography. And he's going to talk about various analytical tools and how these can be used to assess the health of these systems. So in his talk, Elementar Soil and Agricultural Applications, he's going to discuss some of the work from their customers, and he's going to show this using several instruments that have been used within both environmental and agricultural sciences. So over to you, Calum.
Thank you very much. I'll just share my screen. Thank you very much for the introduction. So as Paul mentioned, I'm one of the product managers at Elementar, and my market area focuses around all of the environmental applications. And today I'm going to run through, I think, 4 examples of some work we've done with either customers or in our application lab that focuses on these topics using four different instruments that we manufacture and sell. To start off with, I'd like to give a quick introduction to Elementar, which Paul already quickly introduced. For those who maybe don't know who we are, so as Paul mentioned, we're one of the leading manufacturers of organic and inorganic elemental analyzers, along with other things such as TOC analyzers and stabilized tilt mass spectrometers. And yeah, as you can see here, we continuously sort of try to innovate, work with customers, work with industry to create the solutions that are needed for the research that's being done. So yeah, we have a sort of quite a broad portfolio. So we have the sort of staple business, which is the organic elemental analysis, but we also have a range of inorganic elemental analyzers, nitrogen and protein determination for things such as foods or agriculture applications, TOC analysis. So you'll see from one of the instruments in a few minutes for doing solid TOC, so total organic in solids such as soil, but also a range of liquid TOC analyzers for doing total organic carbon in liquid samples such as river, wastewater, things like that. We also have OES, so optical emission spectrometry, and stable isotope analysis. And you'll see a few of our stable isotope inlets in some of the examples as well. So just a little bit of background about the company. So I'm based in the UK, where we manufacture all of our stable isotope mass spectrometers and the associated inlets. The company itself, Elementar, is German, and we have a headquarters in Langensalbel in Germany, which is just outside Frankfurt, and somewhere around 240 employees worldwide. About half of those work in Germany, and we have about, I think, 30 or so in the UK now as well, and a number at different subsidiaries worldwide. And yeah, we sort of work across a wide range of different key markets. Today, you'll mainly hear about sort of applications within the agriculture and the environmental market, but we also do a lot of work with chemical companies, energy companies, things like forensics, so looking at food forensics, for example, or environmental forensics and materials. So where do our instruments come into soil and agricultural science? So there's a huge number of different sort of application areas where our instruments can help, from sort of more simple things, just looking at a carbon percentage or a nitrogen percentage within a soil or a sediment, but also things such as looking at sort of drought studies and plant physiology studies in crops and plants. So whether that's from a sort of pure research perspective in somewhere like a forest, or a field of crops for looking how efficiently your crops are growing. Nutrient cycling, so looking how nutrients are being used across an agricultural system, or tracing nutrient pollution, so how those fertilizers are getting washed into a river system, and how that's affecting the river system, and tracing where those are coming from and how they're being transformed in the system. And then things like soil organic matter and carbon cycling. So as a huge topic at the moment, thinking about things like carbon credits. So understanding how that carbon is turned over in the soil, and which types of soil, which types of soil are storing more carbon or releasing carbon. And then things like nitrogen fixation studies as well, and understanding that plant environment interaction and how that links together. So I'll start off with the sort of first example, and that's really something that's been sort of quite key to us over the last year. We've been running a huge campaign on soil carbon and biochar actually analysis. So looking at how that soil carbon can be accurately quantified. And why is that important? I'm sure many of the people on this call already know today, but understanding soil carbon can give answers about a huge number of things. So as I already mentioned, there's things such as carbon sequestration and climate. So understanding which ecosystems are having more or less carbon stored or released. Thinking about things like soil health and fertility for agricultural purposes. Water management, so understanding which systems are having problems with how how drainage works and things like that. And there's lots of research being done on the best types of ecosystems for that carbon storage, and water management is a big part of that. Food security, so linking into that soil health and fertility, so understanding sort of what carbon and nitrogen ratios, for example, are best for efficient growth of crops. And then sort of the last one on that list is sort of fairly obvious one, but generally understanding soil carbon gives us a lot of information on the biodiversity in these systems. More biodiverse systems, obviously generally better, and those with more organic carbon in the system quite often are more biodiverse. So just to run into some of our instrumentation. So the instrument that we recommend for doing solid TOC samples, for example, soils or other things like biomass, biochar, solid waste, would be the SolidTop Cube. And this is a temperature ramping system. So rather than having to, for example, run 2 samples where you're acidifying a sample and then running a sample that's non-acidified to get your TOC and your TIC separately, you can run this all in one system. And they run in these small crucibles, and the system ramps up. So you get your TOC released at, for example, around 400 degrees, your ROC, so that residual oxidizable carbon, so that sort of graphitic carbon released around, say, 600, and then your TIC released at around 900 degrees. So within one sample run, you get your full suite of information about the TOC and TIC within a sample. And one of the nice things about this system is that it is built to try and be easy to use. It's reusable crucibles, so you can weigh your sample out into crucibles, load them into the system, and then leave the system running. You can see across the bottom of this slide an example of the crucibles that we sell with this system. So yeah, it makes running samples for TOC a lot easier than sort of traditional methods. And I know many people will be running things like loss and ignition. We actually have some really interesting studies that have been done comparing things like loss and ignition to traditional elemental analysis to this temperature ramp system. And you get some very, very good results off it. You obviously run into issues with loss and ignition because you have a correction factor for it all, and you potentially run into problems when you're running sort of different weights of sample or different sample types. And why is it important to get those 3 fractions? Well, they each tell you something slightly different about the soil or sediment that you're measuring. You have your TOC, which is bioavailable. It's coming from living, from residues of plants and animals, versus your residual oxidizable carbon, and that's much less bioavailable, much harder to break down, and is that sort of elemental carbon, graphitic carbon. And people are thinking a lot about that now with things such as biochar and trying to lock carbon up within systems. And then you have your inorganic carbon, generally carbonates, which is also not bioavailable. And we've been doing quite a lot of analysis with one of our customers in the US, and they're really looking at trying to optimize sample weight versus sieve size of sample. So as you can see from these sort of 2 examples here, if you're not sieving very finely and you're putting a very low sample weight in, your results will inevitably be not quite as good. While if you're sieving to a very, very fine degree and you're putting a lot of sample weight in, then your results potentially be better. But there is sort of a trade-off there with how much effort you're putting in for sort of sample preparation and things like that. And we've been doing a lot of work with them to sort of find this sort of happy medium where you're getting very good data, but not putting a huge amount of time into sort of sieving or prepping those samples. But I mean, looking at this system, I mean, even when you're not really sieving very finely at all, you're still getting quite acceptable standard deviations here. And then I just wanted to introduce one of our customers within this sort of field who are running, I think, 2 SolidTop cubes now. And there's, I think, some information about them on both our website and I think the BS Cube. They've also done maybe a blog post on them as well. So this is Verdant Carbon. They're a sort of startup soil sampling analysis company who have basically focused on testing that organic carbon in agricultural soils to try and allow initially farmers, but also other landowners, and they've been working with a number of different research institutes to try and better understand soil dynamics and think about things such as carbon credits and storing carbon long term. But but obviously these things, they take a while to sort of filter through to things like policy and things like that. But I think customers like this are sort of pushing that policy forward by getting those accurate measurements of soil carbon. So the next instrument that I wanted to sort of talk about is thinking more about sort of soil respiration, looking at sort of greenhouse gas studies using stable isotopes. So we have a number of different sort of potential application areas when we're thinking about looking at greenhouse gases using stable isotopes. One of the sort of most well-known types of study is looking at things like labeled 15N nitrate. So you can see how that labeled nitrate isotope travels through a system as a tracer in agricultural systems. One of the other big application areas is using dual isotopes. So looking at the 15N and 18O, so the nitrogen and oxygen isotopes of nitrate to determine sources of nitrate in the environment. And you can see from the figure on the right-hand side that when you look at both the 15N and the 18O, you can determine where about or where that nitrate has come from. And that's really useful for looking at things such as I mentioned earlier, looking at how fertilizers have been washed into river systems. Where are those coming from? How are they being transformed? So you can see the line on the graph showing denitrification. When you look at both the 18O and the 15N, you can see those types of transformations. So whether there's new nitrate arriving in the system, whether there's nitrogen fixation going on, whether that nitrate's being recycled within the system, things like that. So these isotopes are basically a fingerprint for what's happening. And then something else, such as determination of carbon and oxygen isotope ratios in atmospheric CO2. So looking at how where that CO2 is coming from. And one of the other applications is carbon isotope ratio analysis of atmospheric methane. Again, big greenhouse gas. And the system we do this with is the sorry, IceFlow GHG. So I'll go across from left to right to sort of introduce this system, as it's a little bit more complicated than our sort of standalone Solitop cube for TOC. So we have the software, which is sort of the day-to-day interaction with the system. We have a mass spectrometer for the measurement of the isotope ratios. So that'll be a isoprime vision in this case, but also our other mass spectrometer, isoprime precision, and the preparation unit, which is the IceFlow GHG. So that's where your nitrous oxide sample, your CO2 sample is purified, cryogenically concentrated, and separated. So for example, if you were running nitrous oxide on the system, you need to separate that from the CO2. And then we have an automated sampler for running your samples from septum sealed vials, which means you can basically load the system up and leave it running. So to give just an example of how that would work within the system, and if a sample takes approximately 12 minutes, you start your analysis, you go through the sort of scrubbing process, removing water vapor. Depending whether you're analyzing CO2 or N2O or methane, you go down a slightly different pathway. And then your sample would be purified on a cryogenic trap in liquid nitrogen. And then that sample gets passed from your prep system, so the IceFlow GHG, through to the RMS, and you can get your isotopic regions of your carbon for your CO2 analysis, or your nitrogen and oxygen from your N2O analysis. So just to give an idea of where this instrument will be used in agricultural studies or environmental studies. So we have an example here of a denitrification study. So 15N labeled nitrates used to trace the fate of nitrogen in agricultural systems. So you get N2O being one of the results of the denitrification process, N2 as the N member, but different things that are happening in the soil, for example, can affect how much N2O versus N2 you're getting. For example, things like the C to N ratio can affect this. So be able to look at the the two different gases with the nitrogen and oxygen isotopes, you can help budget that nitrogen through that microbial process. And you can study that within a field or from a microcosm. And you can see here sort of the results of that on the right hand side. One of the other things that you can do with the isotope is plant and soil respiration experiments. And so looking at the carbon isotope rather than the nitrogen isotope. These generally, presented from respiration experiments as Keeling plots. And we basically ran sort of a simplified version to sort of highlight the instrument here. So the experiment started off at night in a darkened microcosm. So you can see where we've got time 0 on that plot. You've got the initial 13C value within that microcosm, and then as the plants respire, that value changes. So you can basically trace where that respired CO2 is coming from. So move on to the next example. So jumping back from isotopes to elemental analysis. One of the other sort of big customer areas we have is agriculture, and one of the big study areas, especially the link between sort of the universities and the agricultural world, is thinking about things like yield optimization. In this example would be grain farming, but within other crops as well. And there's a number of different users who are doing sort of continuous field trials and analyzing results, or looking at the soil that's being that the plants are being grown in using our instruments. So why is it important? So grains are obviously a staple food for many people, and global demand is rising steadily with increased populations, but we generally are losing arable land. So you can see from the right there, from the 1960s to I think 2021, we've lost a huge amount of arable land per person. So there's a lot of customers that are investigating the factors that might increase yields per hectare. And soil analysis, so understanding what they're being grown in, plays an important role in this type of research. And we've got one customer at the University of Gießen, in the Department of Agronomy, Professor Hunnemeyer, who's been running sort of a continuous field trial for a very long time, using a number of different crops. And there's a number of types of questions that he's trying to answer. So how can the use of those different fertilizers affect the carbon content of the soil? And how does that in turn affect crop yields in the long term? And what type of tillages has a sort of critical influence on water retention and carbon accumulation? And he uses one of our elemental analyzers to do this. I'm not sure if it's this one, but he basically looks at the carbon and nitrogen content in his soils and also within the biomass of his crops, and can use a conversion factor on the nitrogen results to sort of calculate nitrogen removal, or look at things like protein content. And that data is a prerequisite for thinking about how he's going to balance the nutrients in crop rotations, understanding how efficiently that fertilizer is being used, and using one of our instruments to do it makes it possible to analyze sort of large numbers of samples very quickly, and then evaluate that data straight away. And so just to introduce one of our elemental analyzers, so we have a broad range of elemental analyzers, basically focused on different sample sizes. So this one here is the Unicube, focused on maybe smaller sample sizes, but we also have a macro cube, which is focused on much larger sample sizes and is generally used in the soil world a lot, and the Variomax cube, which is the sort of largest sort of sample sizes, and that has a similar sampler to what I introduced on the Solitok cube. So you sort of weigh your samples out into crucibles, and that again helps speed up sample preparation. So most of our elemental analyzers look at CHNS, so carbon, hydrogen, nitrogen, sulfur, and then there tends to be options for oxygen or chlorine and things like that. And we use a slightly different way of doing it to some of the other major manufacturers. So we use temperature programmed desorption. So you have a desorption column where your different compounds are adsorbed to. So you have your CO2, your H2O, and your SO2 for your CHS. Your nitrogen N2 doesn't get absorbed. And then those are released at different temperatures. And yeah, fully automatic analysis, so a number of different position sampler options for sort of fast, efficient analysis. And then I'd also like to talk a little bit about tracing carbon uptake in plants from different soils. So we have a really interesting study that was done by Lily Zay from, I think, Dresden University. So there's a few papers on this from 2019 running through to 2022. It's really looking at sort of understanding how peatlands store carbon. So I'm sure many people know that peatlands store about a third of global carbon store on about 3% of the land surface area. So there's a huge amount of carbon being stored there, but obviously with changing climates, some of these ecosystems are changing dramatically, and there's concern that as things warm up and some of the types of plants change from maybe these sphagnum moss to vascular plants with root systems, that the ability to sink carbon to peatlands might be changed, and some of this carbon might be released. So there's experimental setup using, again, similar to how I mentioned in the greenhouse gases, but then this one using 13C labeled water, so an isotopically heavy water, to quantify how vascular plants are assimilating and storing that carbon in the field, and then look at sort of above and below ground carbon allocation within these peatlands, depending on the different functional types of plants and also on the temperature. And there's some evidence that warming temperatures might affect these stores, mainly due to sort of increasing microbial decomposition, but also because the sort of root systems expanding from the vascular plants that basically tap into these maybe more ancient peatland stores. So the system that was used to do this, I'll run through a few results in a minute or two. It's the Isotok cube. So this is a fairly unique system to Elementar, where it's a liquid TOC system that is attached to one of our IRMS. So we can look at the isotopes of dissolved organic carbon and nitrogen as well. So you can see here in a similar sort of layout as I introduced the Isolo GHG, we have the sort of software system that runs the day-to-day things. We have a low concentration module with this system, so that's basically an optional add-on for this system for looking at your nitrogen or very low concentrations of carbon. We have the Isotok cube, which is very similar to our liquid TOC systems, with the sampler on the top for the vials, and then your samples converted to CO2 and then run through the Iceberg Precision IRMS system to get your delta 13C for your TOC. And there's some really interesting results from this study. Kind of expected that, yes, as temperatures increase and these ecosystems potentially drastically change, that we do end up, with fresh stuff being put into the peat. And although some of this is offset by reduced above to below ground allocation rates, it still means that as we add these vascular plants and root systems to peatlands, then these sort of things that are added, these fresh assimilates, are more likely to be respired than translocated to the roots, and they could also potentially mobilize some of that ancient carbon that's stored in the peat. And with peatlands storing such a huge amount of carbon, that's potentially an issue for carbon budgeting in the future. So that's my sort of quick run through sort of a few of our instruments. I'm very happy to try and answer any questions that anybody might have, or at least point you in the right direction to get that answer. But thank you for letting me talk to you for sort of half an hour.
Great, thank you very much, Calum. That was excellent, just going through the range of instruments and also their applications in soil science and also plant and soil science as well. What we're going to do is I'm going to ask you a couple specific questions and then go over to the question board just while people post their questions. We unfortunately couldn't be joined by Charlotte Chivers. She's ill today, so we got a cancellation earlier this morning. But Charlotte is going to be at the annual conference, which is held in early December. And in that, she's going to share some insights from her project on Soil Smart project, and also as part of some of the EU mission soil projects as well. So you will have a chance to engage with her there, and she was going to give a very different angle on our talks on soil health by looking at the citizen science approach. So just to kick off, Calum,
Yep.
The approaches that you showed show basically new abilities to measure properties of soil. And just in general, why do you think it's really important to get some of these quantitative measurements of soil health just to contribute to understanding agricultural systems?
Yeah, it's a good question. I think in the past, a lot of the analysis, or a lot of places have done very small scale studies. So rather than doing a whole field or a whole woodland area, just because you need to do a lot of the preparation manually, and there's a huge amount of time put into that, they've just been done on a lot smaller scales. With sort of the development of some of these instruments that make things a lot more automated, some of these studies can be done on a lot larger scale, which I think makes a big difference. So rather than getting a lot of estimated measurements, you're getting actual measurements. The other thing is, at least from what I've read and what we've seen, is a huge amount of sort of the soil analysis is being done using techniques such as loss and ignition, which is quite quick, but maybe doesn't give you all the information that you could get from, for example, a ramp temperature analysis. And there's lots of things such as there's multiple papers out there using different correction factors for loss and ignition. So maybe even if it's a few percent either way, you're maybe going from a system that's respiring carbon to taking carbon in. So I think the more accurate and precise you can be with these things, the better.
Okay, great. Thanks, Calum. We're going to open up the floor actually to allow people to turn their cameras on if they raise their hands later on, but we'll go through the question box first. So please, if you've not posted a question yet, put it up there. I'm going to start off with one by Julia Cooper, and this is based on the measurements of carbon with your instruments, and whether or not you can use a temperature ramp to differentiate particulate organic matter from mineral associated organic matter purely by running a temperature ramp with your instrument.
So, I mean, it's fairly, so first of all, the system has the flexibility. So if you did want to remove a fraction, you can by acidifying and then running. But I'd say that the temperature ramp really only separates your it separates all your organics because it's only really run off temperature. So you're going to get everything that comes off up to 400 degrees or up to 500 degrees or up to 600 degrees at that temperature. One of the nice things that you can do with it to separate those things a little bit better is gas switch. So when I introduced the system, I don't think I went through that you'd run it on, as a combustion system, so running everything on oxygen, so everything's just burned. But we do basically offer a method that gas switches. So instead of running from using oxygen, you could run using nitrogen. So you could pyrolyze a sample instead, and that does give more flexibility within the system to try and separate things out a little bit differently.
Okay.
One of the things, yeah. So I was just going to say, and one of the things we also allow people to do is create their own methods. So if you think that one type of organic matter is going to come off at a slightly different temperature, or perhaps you think that you can separate that out even more. So generally, most people use 3 ramps. So you get your TOC, your ROC, and your TIC. However, maybe you think you're looking at a soil that has maybe slightly different a different makeup. You split that into 5 ramps and do slightly different things with the temperature and switch the gas at different stages. Then maybe you can separate things out a little bit more.
Okay, great. Thanks very much. If Julia wants to ask another question on that, she can raise her hand later on. Just a general question is, when you're looking at people within soil science using your instrumentation, is what you produce driven by the soil scientist, and is the technique driven by the soil scientist, or is there an adoption where they're basically taking things from other disciplines and applying it to soils?
That's really interesting. I mean, I think, if you look at Elementar's history, we've sort of developed some of these systems with maybe less market input to start with, and then as they've developed, they've had more and more market input into them. And the Solitalk Cube is one that has had quite a lot of market input, I think, from customers and people that want to do slightly different things with it, because it's a fairly versatile instrument. So in particular, when you think about soil scientists, they have slowly moved away, are slowly maybe moving from things like loss elimination to ramped systems or elemental analysis systems. And as they've done that, I think they probably have picked up techniques from other areas. So one of the sort of application areas we sell quite a few Solitalks into is looking at waste. So we have a number of different sort of companies that are looking at different organic waste that use our Solitalk Cube. So they've looked at sort of the methodology, there, and so some of that gets sort of pulled into the sort of other application areas. We also collaborate with people all the time on developing new methods for systems. So you've got the system there, and we're always sort of really interested in people who've got an idea for different techniques to run with that instrument. And method development is one of the things that's, I think, is quite, it's something that we've always got involved in, because as scientists, you tend to know the samples much better than we do. We might know the instrumentation, but you know what those samples, or what you think those samples are going to do, better than we do.
Okay, great. Thanks, Calum. Last one for me is that when we look at ways to assess soil health, there's everything from qualitative field assessments to detailed laboratory assessments. Obviously, the qualitative field assessments allow for very rapid measurements. They're easy to apply. Farmers can apply them directly often, so they're very good for citizen science and letting people learn about their approaches. But we often assume that if it's measured by an instrument, it's correct. And I know that resurvey of a country I won't name, of their soils, where there's billions being invested, there is a lot of data that's being generated that has got quite poor quality control over it. So it's quite interesting to hear that just in your TOC analysis, just the sieving of the soil can affect the result. What I want to know is, first of all, why does this occur? Because you're just combusting the whole sample anyway. And what approaches would you recommend to allow for agreements between operators who may have instruments placed in different laboratories.
Yeah. So yeah, again, and this is something that's not just soil focused. This is completely worldwide for almost every different application. Is that people use the same instruments and get slightly different results. And the answer is generally that we need sort of more standards, more norms that people follow to the letter, really. I mean, there are quite a few that are associated with soil now. There's the sort of EN 17505 for ramped analysis. So that gives very sort of strict layout of, right, this is your TOC 400, this is your ROC 600, this is your TIC 900, because that is something that potentially causes differences in results, is whether you're getting full conversion of your sample, and whether you're running at the same temperature ramp as somebody else. And one of the things that I'd say that we do see slight differences with is, and I think it's quite interesting because I think we'll probably be presenting on this at your December meeting, is if you look at, for example, a loss and ignition sample, a acidified prepared TOC sample, and then a temperature ramped TOC sample, there are potentially differences there. And it's firstly understanding why there are differences. There's 3 different methods, and they can have different results because of those things. But I think if you're talking about say for users using, say, specifically our Solitop cube, they agree quite well. And if customers should talk to each other, that's another thing. I mean, we speak to our customers all the time and try and introduce them to each other. So if somebody's doing something slightly differently, maybe the better way, we can highlight that to everybody in the future. But there are differences in the way people measure things, and it's not an easy fix, I don't think, especially if people are using very different techniques, especially between different countries as well. I mean, I understand that there's different standards in different countries for different things. We're seeing now that this temperature ramping norm that's just come out has just been turned into an ISO norm. So it first got turned into EN norm, so it was just European wide. It's now been turned into an ISO norm, and I think things like that help.
Okay, excellent. Thanks very much, Calum. What I'm going to do now is allow people to raise their hands if they'd like to ask anything with their cameras on, and then Dom Sims can turn your camera and microphone on. So if you've got any comments or questions regarding soil health, it doesn't need to be totally related to the presentation. We'd gladly hear them. So I'll see if anything comes up. Just as we wait for that, someone has posed a question about whether or not the talk will be shared, and all of our Zoom into Soil seminars are shared online, and you can access them via the soils.org.uk website.
And if anybody's interested in more detail about any one of those studies, then feel free to get in contact with me, and I'll happily share the paper that's been quoted or any more instrumentation details that they'd like to know.
Okay, I'm not seeing any hands raised unless Dominic can tell me otherwise. So what I think we'll do then is, unfortunately, Calum had to cover for a longer period of time. And there is one more question that's come up. So this is from Karam Shahzad. Can you please tell of any comprehensive soil assessment model developed by your organization? So to answer that, it's Elementar is an instrumentation company, so that would not be within their remit. British Society of Soil Science as well, we support the study of soils. So there will be many comprehensive soil assessments developed by our members, and those can all be found within literature, but we do not advocate for one single comprehensive soil assessment model. Okay, so I think that's probably it for questions for the seminar today. So I want to thank Calum Preece for his excellent presentation. Also, I wanted to thank Charlotte Chivers for volunteering to present today, and it's a shame that she was unwell. Okay, so that's it for the seminar today. So thank you very much, and we look forward to welcoming you to the next Zoom into Soil.